All right, thank you for the introduction, for setting that up, Pat. So today I have the pleasure of introducing friend and colleague, Dr. Myp Sekhon. Dr. Myp Sekhon is a neurointensivist at Vancouver General Hospital and a clinical assistant professor in the Faculty of Medicine at the University of British Columbia. He completed his medical training at the University of British Columbia and then traveled to Cambridge to work in Addenbrooke's Hospital under the supervision of Professor David Menon, where he acquired his neurocritical care specialty. Here he learned new techniques such as invasive neuromonitoring, and he brought those techniques back to Vancouver General Hospital, where he's pretty much built an invasive neuromonitoring program from the ground up, and is now leading some very, very interesting work into the pathophysiology of hypoxic ischemic brain injury in cardiac arrest. And what I think is very, very interesting and relevant to a lot of us in the field of cerebrovascular physiology with Dr. Sekhon's work is the blend of physiologic work as we see it day to day with the clinical aspect as well. And so I think we'll find a lot of the aspects of his work that he'll talk about today quite interesting. With that, the title of his talk is Clinical Insights into Ischemic Brain Injury Pathophysiology in Humans, and I'll let him take over.
I wanted to just acknowledge, so first off, thank you very much, Ryan, for the introduction. I come here very humbly. My primary training is as a clinician, but I must say my view on medicine changed tremendously in 2016, I should say, when I met Phil Ainsley in Kelowna, and I met people like Tony, Bain, and as well as Ryan Hoiland, who've changed how I view medicine, and I think really showed me what is possible and what we do not know in clinical medicine. And so it's been a real pleasure, and I'm very excited to show you some of our work that has emanated straight from that. All right, so what I'll talk to you about today are the insights of some of our work that we've looked at in terms of cerebral ischemia reperfusion and the physiologic context of that, and how we think that what we're learning from this translational research program may have implications for identification of future efficacious therapeutic strategies and resuscitation management. Before I start, though, my disclosures are that these organizations do help fund our work, but I have no industry sponsorships, and so however uninformed my opinions are, they are truly my own. Hypoxic ischemic brain injury following cardiac arrest is a global ischemia reperfusion injury of the cerebrum and the adjoining structures. And in those who are resuscitated successfully out of cardiac arrest, out of circulatory arrest with cardiopulmonary resuscitation and therapeutic interventions thereafter, this is the primary determinant of outcome in excess of 90% of patients. So patients die of their hypoxic ischemic brain injury. And regrettably, it's remained a relatively elusive target in medicine. The outcomes associated with HIBI, as we term it, are dismal. As I said, roughly of those survivors who do have return of spontaneous circulation, as we call it, only 10% of them go on to survive in the most severe states. And of those survivors, there's significant neuropsychiatric sequelae long term that are related to the pathophysiological mechanisms that are occurring in the post-resuscitation setting, we think. And traditionally, we've had therapeutic hypothermia that's been championed as a mainstay management treatment for this devastating disease. And even now, recently, just this past year, from a really landmark trial from Sweden, that appears that it may not be as efficacious as we thought it once was. So really making it imperative upon us as a medical community to look at this disease in more detail to identify potential other therapeutic avenues that we should study. Now, when we go down to the level of the neurovascular unit, obviously something that all of you know in much greater detail than I, But we've got a healthy astrocyte encasing the cerebral vasculature. You've got surrounding neurons with dendrites and axons, lots of glucose, lots of oxygen. In the ischemia reperfusion setting, largely based off of animal studies, due to the relatively inaccessibility of brain tissue in the acute hospitalized setting, we think that there's many different pathophysiologic mechanisms that are occurring, some of which include microvascular thrombi, a porous blood-brain barrier. You have depletion of glucose, so neuroglycopenia, cerebral hypoxia or brain hypoxia, ultimately culminating in metabolic stress of the neurons and then subsequent death thereafter. These pathophysiological mechanisms haven't been, they've been postulated to occur for the better part of 20 to 30 years, although significant breakthroughs within the in vivo model of humans have not yet been made. And we think that we've hopefully opened the door to looking at this in further, and I'll show you that in a little bit. Now, based upon this pathophysiology, clinically, what we tend to do is we tend to think of this disease relatively simplistically, if I'm being totally honest. There is the primary injury that occurs upon arrest of the circulatory system for which we provide CPR. Immediately upon return of spontaneous circulation, recovery of the innate cardiopulmonary hemodynamics, then there's a secondary injury that occurs. And that's what's modifiable for us in the intensive care setting. We theorize that that secondary injury, the majority of it, is attributable to lack of oxygen within neural tissue and then subsequently necrosis. And so as such, there's been dogma in critical care for decades that, well, we ought to balance oxygen delivery and improve utilization. The utilization aspect of this, sort of came up with all the literature in and around therapeutic hypothermia for years. But the oxygen delivery part of this teeter-totter has only recently come up in the past number of years. This next slide, I'm almost embarrassed to show because I typically give this talk to clinicians, and so I have to walk them through this a little bit. But just suffice it to say that in the setting of HIBI, in the setting of post-resuscitation neural injury, we know that the majority, most of these patients aren't anemic. We can often keep a fairly accurate saturation of hemoglobin using the ventilator. The majority of what's thought to be responsible for the additional ischemia reperfusion injury and cerebral hypoxia is related to reductions in blood flow. And the two clinically relevant markers or variables that we can modify relatively easily are arterial carbon dioxide tension, and of course, mean arterial pressure. In the setting of HIBI, most HIBI patients have relatively low ICP. There are the extreme cases, which are probably not salvageable to begin with, making the clinical marker of mean arterial pressure as the most modifiable and relevant variable in terms of determining overall oxygen delivery. Now, again, this is relatively simplistic. And so when authors and clinicians have looked at this, there's been a number of observational studies that have been published to characterize the problem, none of which I'm going to really show you in detail, other than to suffice it to say, many different groups have looked at this, and there's been more studies repeated on this that have associated low blood pressure, low mean arterial pressure to a worse outcome. But it's hard to know if that's just a manifestation of the overall severity of injury, or if in fact there is reduction of cerebral blood flow, cerebral hypoxia, or brain hypoxia, and then subsequently deleterious pathophysiologic mechanisms thereafter. One of our medical students, Tahara, did a systematic review a number of years ago. And again, we concluded that among all these observational studies, each of which are flawed for different reasons, there are improved neurological outcomes that are associated with higher blood pressures after cardiac arrest. But this is probably the most important line. In fact, there's a need for further research. Well, thankfully, since then, there's been 2 randomized control trials that have been conducted. Really elegant. This is the first one by Amalut et al. 112 patients underwent randomization. randomization to what we target as a normal map in critical care, so 65, for patients using exogenous vasopressors like norepinephrine and epinephrine and so forth. And then what they termed an early goal-directed hemodynamic optimization group. So mean arterial pressure of 85 to 100. And the primary outcome in this study, though, was the ischemic burden on MRI. And they showed separation of the groups with respect to mean arterial pressure. Not surprisingly, much more norepinephrine use in the high MAP group. I'm not a big believer in NIRS, but and so this wasn't terribly surprising to me. But further off into the after return of spontaneous circulation, no different in difference in cerebral oximetry. And in a subgroup of a small number of patients showed that the flow of the MCA, the mean flow of velocity in the MCA was higher in the high MAP group. Regrettably, though, they did not demonstrate differences in the ischemic burden on MRI. So there's a total disconnect. What we think is the most important physiological variable, mean arterial pressure, augmenting it doesn't lead to less ischemic burden on MRI, nor did it have any real signal. It wasn't powered for clinical outcome, but didn't have any signals for improved clinical outcomes. This was followed up by a 2x3 factorial design trial called Coma study, published out of Finland. Again, phenomenal achievement that this group undertook. 123 patients randomized, again, to high MAP versus normal MAP. They showed very good separation of the groups in terms of their mean arterial pressures in both patient groups, in terms of the exposure of the variable to both intervention groups. Their primary outcome were biomarkers, and specifically neuron-specific enolase, which has its inherent problems from extracranial production, but did not demonstrate a difference. Further to that, though, they also didn't show a difference or a signal in clinical outcomes. So the question here is, well, what gives? We've, in clinical medicine, preached that mean arterial pressure and optimizing oxygen delivery is so important in this disease entity in ischemic brain. But then actually undertaking some interventions to optimize that aren't associated with better biologic or clinical outcomes. So the next question we had was, well, are there missing links in the pathophysiology? Are we as clinicians viewing this disease far too simply? I think the answer to that is yes. And so I'm going to take you through what we've sought to do to try to shed light on this devastating clinical entity. Based on what we learned and what I was taught in the UK and coming back to Vancouver, we decided the first problem is we don't understand this disease in humans fairly well. And what better way to do that than to use some of the monitoring devices to characterize the physiology of these patients, the cerebrovascular physiology of these patients, in an in vivo human model with disease. And so what we end up doing now as part of, in many cases, standard of care, but an ongoing prospective interventional research project, is we make a frontal burr hole. So it's about half a centimeter, less than about 4 millimeters in diameter. We pass a cranial bolt through it, so a double lumen styleted tube, and then pass 2 catheters. The first catheter is an intracranial pressure catheter. So that's just reading in millimeters of mercury, the pressure in the intracranial vault. The second catheter is a Clark electrode, which reads the dissolved tension of PO2 in the brain tissue. So P, meaning pressure, and then BT, brain tissue, oxygen tension. If you were to take the tip of that catheter and put it into the left ventricle, it would continuously read your PaO2. And so this is now done as part of standard of care. The insertion of the bolt is done on the non-dominant side, so usually on the right hemisphere, and then the catheters are situated in the subcortical white matter. That's not really modifiable, unfortunately. So we're getting subcortical white matter focal information. Now you might say, well, how about in the rest of the brain? Well, the reason why we can't put these catheters in other more viable or important pieces of tissue, the thalami, the basal ganglia, and so forth, is the risk of course, hemorrhage and damaging really critical areas. So that's why we place it where we do. Now, in the setting of a diffuse injury, like global hypoxic ischemic brain injury, we are taking a leap and extrapolating that whatever is happening here is occurring in the rest of the brain as well. So there is that methodologic leap that we are taking. We also put, as part of the initial study, near-infrared spectroscopy bilaterally in the forehead. I'm not going to show you data from this because it was wildly disappointing. Suffice it to say, I'm not sure in the health setting, but in the disease setting, particularly following cardiac arrest, near-infrared spectroscopy is unable to provide any sort of reasonable measurement of or estimate of brain oxygenation or cerebral hemodynamics. And so this was disappointing to us, but a negative finding is of course a very important finding. And then also we place a retrograde oximetric 15 centimeter line into the dominant jugular vein and then situate it at just distal to the sigmoid sinus in the jugular bulb. And so this will read continuous saturation of hemoglobin as it exits the brain. So because we have an arterial line, then we have cross cerebral circulation ability to sample biospecimens for serum and plasma, and then conduct analyses based on that. Also AJDO2 differences. All of this information, and this is, I think, quite a strength. All of this information goes into a software program that records it at 300 hertz. So tremendous granularity of data to look for higher order relationships between these variables. And then, of course, we do collect core temperature and then end-tidal carbon dioxide tension continuously. Now, more recently, and this is, I think, far more exciting, we've added two additional catheters to this monitoring platform because, of course, two is not enough. So this is what our quadruple lumen bolt looks like, placed in the same place. This is the PBTO2 catheter right here that's passed in one stylet. We have the intracranial pressure catheter passed in another stylet. We have this catheter here, a microdialysis catheter. So through the principles of dialysis and movement of molecules across a semi-permeable membrane, we infuse an isotonic fluid into one of the lumens of this catheter, and then it comes back out through a pump, and then we're able to sample it at this port right here. And so at the end of every hour of monitoring and flushing of the fluid, we're able to collect 1 mil to 1.5 mil of what we call microdialysate. Now, as part of our clinical care, we measure cerebral glucose, cerebral lactate, cerebral pyruvate. We get a lactate to pyruvate ratio as a crude estimate of overall metabolic stress of the neural tissue. We also get glutamate and glycerol. Now, more recently, though, we've started, we're very keen on looking to see if we can measure other analytes as part of this, of these samples. So that gives us some metabolic information. And then we have a thermal dilution probe here called the Bowman perfusion catheter, which provides peel or real-time blood flow at the level of the peel arteries in the range of mils per 100 grams per minute. So continuously. So all of this information now is coming continuously. Because we have CBF continuously now, AJDO2 differences, we can monitor CMRO2 continuously. We can monitor cerebrovascular resistance continuously with the CBF and the CPP that we have from our arterial line minus the ICP. So all of this this data, I think we haven't analyzed this next cohort of patients, but all of this data is I hope will shed very important light on this devastating disease. And then this on part B of the image right here, you've got this part of the titanium bolt sits within the skull, and then the catheters are passed through. The Camino catheter, which is the ICP catheter, sits a little bit more superficially, whereas the other three are inserted directly into the parenchyma. This is kind of what it looks like at the bedside. So we have a ton of monitoring equipment. We also place a large intravenous catheter into the femoral vein that sits into the inferior vena cava, and has some external or some serpentine lumens around the catheter that are constantly infused with cold saline, and so we're able to cool the body centrally and maintain normal thermia. We have our PbTO2 catheter or monitor here, our ICP monitor here. You can't see the Bowman perfusion probe in this patient, but then our bedside laptop with ICM Plus, and then all the rest of our monitoring equipment. Microdialysis analyzer is here, and so at the end of every hour, the bedside nurse will take the little collection device, place it into here, and then we'll get our readings for those metabolic variables that I told you about. So as part of this study, I think when we have a problem in medicine, the first thing to do is to characterize it. And so the goal of the first series of studies was not to go too far, but to just get a lay of the land, to plot the raw relationships of these various variables against one another to see what we can ascertain. And so we had two very simple goals. One was to characterize the burden of brain hypoxia. Is brain hypoxia, secondary brain hypoxia in the post-resuscitation setting, even a problem? Are we barking up the wrong tree? Second, characterize the relationships between brain tissue oxygen levels using the catheter and mean arterial pressure over time. Those two trials were negative. So does augmentation of mean arterial pressure even lead to and equate to improved brain oxygenation? And so when we look at the first parts, or just this graph here, where we plotted the raw PbTO2 of all the patients on the y-axis and the mean arterial pressure on the x-axis, it doesn't look like a lot of patients. There's just 10 here. However, given the granularity of our data, this is a culmination of 25 million data points. We found a couple of interesting things. First, as part of ethics to do this, we cannot blind our colleagues to this information. We're doing an invasive procedure in a very sick patient. And so as part of this study, we provided our colleagues with access to the information so they could at least intervene upon it to try to correct the physiology if it was abnormal. What we ended up finding was, despite myself and my colleagues trying to optimize brain oxygenation using what we know from traumatic brain injury, we were unable to, in upwards of 40% of the monitoring time. So no matter what we did to CO2, transfusions, which Ryan will talk to you about in a little bit, augmentation of mean arterial pressure, cooling the body, there's 40% of the time across this entire cohort, and this is held steady in the subsequent patients, we are unable to achieve normal PbTO2. Secondly, when we plot the raw relationships of PbTO2 against MAP across the whole cohort, there's a positive relationship. Well, that's not that quite interesting. What's more interesting is when you look at each individual line, there's tremendous heterogeneity within patients. And again, this is held true in subsequent patients as well, with respect to their individual relationships of PbTO2 and mean arterial pressure. So if you look at the one patient on right here, certainly as mean arterial pressure is augmented, the PbTO2 takes a sharp rise, whereas this patient doesn't seem to make that much of a difference. The number of 20 is especially notable, because if we extrapolate from traumatic brain injury literature, where investigators have done PET scans at the same time neuromonitoring is in place, the number of 20 is especially notable because that denotes brain hypoxia, an episode of brain hypoxia. So when you look at this data, those are the two things that really are remarkable. We're unable to achieve brain normoxia in some people, and further to that, there's tremendous heterogeneity in the individual relationships. Now, I've just talked to you about one aspect of the oxygen cascade, convective delivery, as Ryan always terms it, and specific focus on mean arterial pressure. But are we thinking about this too simplistically? We make a massive assumption that when we deliver oxygen to the brain, it gets into the parenchyma and it is utilized normally. And so we focus a tremendous amount on convective delivery because those are the variables that we have access to at the bedside. How about diffusion? Is diffusion from the cerebral circulation into the parenchyma normal or not? Well, when we go back to this slide that I showed you, or this graph that I showed you before, when you look below 20, there's a ton of data down here. When you look above 20, there's a ton of data up here. And so there appears to be almost two distinct cohorts: those who are normoxic, who have brain normoxia, and those who have persistent brain hypoxia. When we look at and plot the CPP against the jugular venous saturation of hemoglobin as it's exiting the brain, so at the jugular bulb, so this should say SJVO2. Not part of this is a typo. It's not SaO2, of course. What we end up seeing is there's a cohort of patients here with really high saturation of hemoglobin as it exits the brain, and then patients who have a relatively low or normal saturation of hemoglobin as it exits the brain. Well, in fact, these are the patients who have brain normoxia. These are the patients who line up exactly with those who are persistently hypoxic. So what that means is that there appears to be an offloading problem. So our assumption that as we, from the cardiopulmonary system, manipulate the macro variables associated with convective oxygen delivery, our assumption that those clinical interventions lead to normal oxygen diffusion from the microcirculation into the cell is probably not true. Now, to numerically represent this relationship, because of the principle of continuity, we hold PaO2 very, very stable, so 80 to 100 millimeters mercury in these patients. So we ventilate them very strictly to that. But because we have jugular venous bulb sampling and we have continuous PbTO2 sampling, you can numerically represent the diffusion barrier of oxygen by subtracting PV minus the PbTO2. And this comes from PET scan studies that David Menon has done years ago. And so that gradient represents the diffusion gradient. Now, a reduced gradient means, of course, more oxygen is being offloaded from the circulation into the cells, whereas a high gradient, so a high PV and a low PbTO2, would suggest that there is an offloading problem. And when we look at patients now divided up with those who were normoxic, right? So these are just individual patient numbers. We've got 7 patients who have brain normoxia, 7 patients who have brain hypoxia, so PbTO2 less than 20. The individual gradients are far greater in those who are hypoxic. And this is despite the macro variables being the same, CPP the same, PaCO2 the same, hemoglobin the same. So the macro variables that we associate as estimators of overall convective cerebral oxygen delivery are the same between both groups, but the gradients are far greater, meaning there may be a problem in the microcirculation. Perhaps more interestingly, as we vary CPP in the normoxic group and vary CPP in the hypoxic group, across the group, that relationship decreases. So as CPP increases in those patients with brain normoxia, the gradients decrease. But in the hypoxic group, there's persistent brain hypoxia no matter what you do. Again, suggesting that there's an uncoupling between macro convective delivery and actual tissue oxygenation. On an individual patient level, this is kind of what it looks like. So we've got one patient here, mean arterial pressure plotted on the y-axis. Here's 90. We augmented it up to 100. It went down to 60 for some reason, back up to 90. But you can see here the PbTO2 basically doesn't change. It's got maybe a millimeter of variation, not very much. Conversely, here's a different patient where it starts off at 90, goes up to 100, varies, and the PbTO2 in this person almost mirrors what is happening with the mean arterial pressure. So these patients represent very distinct and unique phenotypes. The clinical implications would be, of course, well, in those MAP trials I showed you, this patient would have of no physiologic benefit of augmenting mean arterial pressure. This patient definitely would. So how do we separate out these patients? How do we identify them in real time so that we can maybe, instead of a one-size-fits-all approach, personalize the therapy? And so the data I've showed you thus far suggests that that leap of faith that we make in clinical medicine between convective oxygen delivery leading directly to normal diffusion utilization just appears not to be the case in all patients. Now, how about the impact of this, though? How about the impact of what this secondary brain hypoxia actually means? So we've identified a problem. The next issue is, does it actually matter? Does it, like, intuitively, I think it does, but does it matter to additional tissue injury and an overall outcome? So this is really, this shouldn't say in review, this has been published just a couple months ago by Ryan. And so although I'm presenting this data, I very humbly want to acknowledge that Ryan Hoiland is entirely responsible for this in a good way. But so he developed this study from start to finish. I'm merely on the paper, but did very little work. So what he ended up doing here was taking our cohort of patients, dividing it up, the post-arrest patients, so the HIBI patients, 18 of them, into those who had brain normoxia, so HIBI NX normoxia, and those who had brain hypoxia, HIBI HX. And so there's that line of 20 that is particularly notable as the cutoff. And then paired this with control samples in 14 young healthy male controls from Phil Ainsley's lab. And so what we ended up doing here was, because we had arterial lines in place, a jugular venous bulb in place, effectively we've isolated the cerebral circulation in an otherwise very systemic disease. When these patients are resuscitated from cardiac arrest, there's tremendous, not only ischemic reperfusion injury to the brain, but also the other vital organ function. And so just getting systemic samples for biomarker analysis, you'd be hard pressed to make the argument that there wasn't dysfunction in the other vital organ systems contributing to that, to those findings. And so by having the arterial and the jugular venous bulb, we're able to draw gradient analysis and to ascertain what is actually being released or taken up actively by the brain during circulatory transit. And so some of the markers we wanted to look at in these two patient groups were markers of injury to the various elements of the neurovascular unit. For astrocytes, glial fibrillary acidic protein. And so what he's done here was, by taking those paired samples in controls for GFAP, in HIBI normoxia and HIBI hypoxia, these are the veno arterial gradient, so a positive gradient, meaning that there's active release in that moment of time. In those patients who are hypoxic, have brain hypoxia, far greater release of glial fibrillary acidic protein, and really no difference between the HIBI normoxics or the control patients. When we look a little bit further and we look at axonal injury, so total tau as well as neurofilament light, again, even more marked results. So big time release of neurofilament light and total tau in those only who have brain hypoxia. And then finally, in terms of cell body injury, a little less impressive, but still pretty marked. So UCHL1, ubiquitin carboxyl hydrolase L1, and neuron-specific enolase. Again, significant differences compared to controls in HIBI normoxics. So what this data suggests is that brain hypoxia, secondary brain hypoxia in a post-resuscitation setting, is associated with de novo injury to the neurovascular unit. Whether or not that's just a sign of injury severity in the initial injury, is something really important to ascertain. So what's the mechanism of this? The first question is this just related to the overall global ischemic reperfusion injury of all the organs? And is that what we're capturing? Interestingly, doesn't appear to be the case. So when we look at these two groups and we look at systemic variables and systemic biomarkers of global injury, like serum lactate, LDH, lactate dehydrogenase, overall pH, serum creatinine, a marker of course, acute kidney injury in the ischemia reperfusion setting, there's no real difference at all in terms of however we look at this, upon admission, peak, or the median values during their admission. Further, bicarbonate levels is a measure of overall acidemic or acid production, troponin levels, AST and ALT, which are markers of hepatocellular ischemic reperfusion injury. Again, no difference. So this appears to be a brain-specific finding, which is very interesting in an otherwise multi-system disease. When we look, now these are small numbers, of course, and you also look at just individual rock curves for the brain biomarkers in terms of detecting brain hypoxia versus the other markers, the more global markers, really not impressive at all. You might as well flip a coin to predict brain hypoxia, whereas the novel brain biomarkers certainly do reflect that cerebral physiology. Next question I have is, so it appears to be a brain-specific finding, but what is the mechanism by which brain hypoxia leads to additional injury? Our thinking in critical care is usually fairly linear. So I showed you a bunch of observational studies, or the titles of them anyways, that suggest that low blood pressure equals bad outcome. So then our solution is to do trials to increase blood pressure. Historically, we've, with the targeted temperature management, high temperature equals bad outcomes. So then the solution is deep hypothermia. Instead of fixing vital signs, though, perhaps in critical care, what we need to do is think about this in a more non-linear path. Just because we have brain hypoxia, does that mean fixing oxygen levels is going to lead to better outcome, or is the pathophysiology non-linear? Does brain hypoxia incite some other kind of injury, which may be modifiable, and then lead to neuron injury? So we wanted to look at this a bit further, and so specifically with respect to the blood-brain barrier, is the blood-brain barrier involved in this? Is there lots of problems with interstitial edema that may compound this? And then further, are there elements of the cellular cell body injury, specifically mitochondrial injury, which could be implicated? First, with respect to the blood-brain barrier, we looked at a bunch of different markers. So both arterial samples and then also arteriovenous gradients. Suffice it to say, we didn't really see very much. So no matter which way we looked at it in terms of all of the various markers that we looked at, and we looked at a bunch of them: Eselectin, P-selectin, soluble ICAM3, Syndecan1 as a marker of the health of the glycocalyx, von Willebrand's factor, thrombomodulin. There weren't really marked differences in the patient groups versus controls. Now, that might not mean that endothelial injury is not part of this. It probably is. We may just have missed temporally the sampling of these markers and our biospecimens to capture it. It may be that there is a very early endothelial injury, but it doesn't appear that endothelial injury continues to occur in the hours after successful return of spontaneous circulation. One of the other things that we decided to look at is these patients end up getting quite a bit of cerebral edema. In many cases, that can be from cytotoxic edema, interstitial edema. And so one of the things that we do as part of routine care in some cases is provide these patients with hypertonic saline as an osmotic therapy to dry out the brain. Very interesting, as we look at the administration of hypertonic saline pre and post, the PbTO2 increases. The MAP doesn't really change. The ICP doesn't really change. The CPP doesn't really change, but the PVO2 almost drops, but the gradient certainly drops. So the implication here is the administration of hypertonic saline doesn't really seem to be changing macro variables, but is there an effect at the level of the microvasculature where we're drawing edema out of the brain, and is that somehow helping improve diffusion? Now, we don't know if that leads to decreased injury, but this finding is specifically in patients with brain hypoxia. When we look at the patients with brain normoxia who've achieved or received hypertonic saline, now there's only 4 patients. We haven't included any statistics as part of this. The findings are not nearly as remarkable as it pertains to the change in the gradient. So this appears to be a change only in those who are having active brain hypoxia. This is one of my favorite slides, and this is from my David Menon's critical care medicine paper in 2004 on diffusion limitation of oxygen delivery in traumatic brain injury. For me and for many in the field, a really iconic paper, where what they ended up doing was varying blood flow by inducing mild hypocapnia and then doing pre and post PET scans to look at the efficiency of diffusion in health and disease, and essentially determined that in many traumatic brain injury patients, abnormal diffusion at the level of the microvasculature is a thing, termed diffusion limitation. And then subsequently took brain biopsies from those patients and electron microscopy. As you can see here, here's a normal neurovascular unit under EM, and then an abnormal neurovascular unit. All this white here is a tremendous amount of perivascular edema. There's microvascular collapse, and this is probably a detached pericyte. So, one of the questions that we have in our upcoming analyses now in this next cohort are, do the pericytes have a role in the abnormal microcirculatory hemodynamics in this disease entity? All right, well, let me just back up. And I'm just going to leave you here now with a little bit of a teaser. I've showed you that hopefully I've showed you that secondary brain hypoxia in this disease entity clearly appears to be associated with de novo injury to the neurovascular unit. The question is, well, what's the mechanism? Doesn't appear to be ongoing blood-brain barrier damage. It could be mitochondrial injury, which we'll have to wait for our next set of analyses. Maybe some interstitial edema, but is there another roundabout mechanism? One of the other things that we decided to look at was some markers of inflammatory or neuroinflammation, and this was more just a fishing expedition, but we found something interesting, and subsequently we found something more interesting. So interleukin 10, this is a relatively anti-inflammatory cytokine, no real difference. TNF alpha, no difference. But interleukin 6, there appears to be active release in those patients who are brain hypoxic. We've now subsequently, it looks like we replicated these findings with interleukin 1 beta. So the question of, does brain hypoxia incite a neuroinflammatory cascade? Does it incite a immunopathologic injury to the parenchyma? And then secondarily, is that modifiable? Those are the next questions I think we're very keen on looking at in the future now. Where is this interleukin 6 coming from? Well, one of the places it can be coming from, of course, are astrocytes or the microglia. And when we look at just a schematic of what we think might be happening is as that injury occurs in the ischemia reperfusion setting, some of the things that we're very interested in now coming up are the differences in the complement factors as a key pillar of the innate immune system. We do know that in ischemia reperfusion models that monocytes do infiltrate tissue. Is that occurring in the brain? It is the autophagocytosis that occurs once these differentiate into macrophages responsible for the neurofilament light and tau release. And then should we be targeting that as a therapy, or is this adaptive? is this what should be happening? So those are some of the key next questions that we're very interested in, and specifically, what is the role of the microglia in this case? I'm just going to show you this really quickly. This is just a peripheral sample to show how we're going to look at this. So this is CyTOF, so mass spectroscopy. A colleague of ours, Kelly McNagny, is a very dear collaborator who is able to identify very rare populations of monocytes in hematologic samples using a screen of 31 to 35 antibodies. And what kind of really made us interested in this was the Eselectin. So Eselectin values were significantly higher in patients with brain hypoxia, and Eselectin being a major endothelial adhesion molecule for monocytes. So this is some of the next steps are to characterize if these monocyte populations are actually reduced in the peripheral circulation, meaning that are they infiltrating the tissue, and should those be targeted in the future? I'm going to stop there. But I hope I've given you a little bit of food for thought, talked to you about distinct pathophysiological phenotypes as it pertains to oxygen diffusion, hypothesis generating in terms of what the causes for this are. Are the astrocytes involved in this? Is there physical widening of the diffusion barrier? Is that even a thing? Mitochondrial injury, stay tuned for that. And then whether or not there's an immunologic injury that is occurring, an immunopathologic response, or whether it's adaptive. So future next steps, we've got wonderful collaborators to work with, people like Leonard Foster, who's going to help us with proteomics to evaluate the intracellular pathways of these of the biospecimens that we've stored in these patients. We're also now getting brain biopsies on these patients as well. And I think we really need to work to distinguish these phenotypes in real time. And then are there considerations for other diseases of cerebral ischemia? I think all of you on this call, why you're so inspiring to people like me is because cerebral, as in clinical medicine, we have gotten much better in many different facets of acute disease. The outcomes of lung injury, shock states, acute myocardial infarction, they're much better. But ischemic brain injury continues to be a massive problem in our society. And much of the work that I think we are doing and that we should do in the future is based upon the rigor and the work of what many of you do in the setting of health. So it's been a real pleasure to be here. I thank you for the opportunity. And again, very humbly appreciate the chance to be here. Just thank a few people. Ryan, it's really been a big inspiration. It has taught me as much, if not more, than Phil Ainsley did during my PhD. Denise Foster, our research coordinator, Rebecca Gray, and then my friend and colleague, Don Grisdale. I couldn't find this emoji is actually better looking than him. And of course, Phil, who really opened my eyes to what is possible, and really, I think what we should be doing in this critical care population, which is delineating the disease pathophysiology before embarking on widespread therapies. Thank you.
Awesome. Thank you for that talk, Mip. So now we'll just take a couple minutes. I see a few questions in the chat. The first one is from Patrice Broussard. He says, exciting talk. Many thanks for that. We know what is the influence of hypoxic ischemic brain injury on cerebral sympathetic nerve activity. Could cerebral SNA overactivity explain, perhaps at least partly, why elevations in mean arterial pressure don't lead to better outcomes in these patients? Yeah,
such a good question. The short answer is I don't know. The longer answer is I'm not sure how. I'd be very keen to hear your thoughts on how we could figure this out. We have there's a bunch of confounding in our patients that I think might present some problems because they are pumped full of so many exogenous vasopressors and catecholamines, epinephrine and norepinephrine, and then sometimes they're getting beta blockade and alpha blockade in various times. So I'd be very keen to, it's something that we consciously did not look at for those reasons, but I'd be very keen to hear your thoughts and how we could or should look at this.
I don't know if you want to.
Could be afterwards or now. Or,
yeah, we could definitely have a chat afterwards for sure. Thanks.
Awesome. We have another question. It says, sorry, I missed the part about how the samples were obtained. Where do the markers come from, blood or CSF?
Yeah, great question. They're serum and plasma. Simultaneously drawn from arterial line and jugular venous bulb. In this next cohort of patients, so we're doing the same thing, but we also have CSF saved from this next cohort of patients. So we'll have arterial inflow, venous outflow, and then what's in the brain, and then in select patients, we are getting brain biopsies as well.
And we'll just take the last question here. Would you consider to perform contrast enhanced imaging on patients to scan blood-brain barrier damage and compare these results with the PbTO2 values? Yeah,
that's such a good question. You know, particularly with the advances of MRI, it's something we're very interested in. The one of the difficulties with our experimental setup is our bolt and some of our catheters are not MRI compatible. So that represents a big barrier to getting these patients down to MRI. Secondly, many of these patients are so critically unwell that even small little movements can elicit catastrophic deterioration. So to take them down from a very controlled setting in the intensive care unit to a relatively uncontrolled setting in MRI is not unfortunately terribly feasible. One of the things we have looked into recently is the advent of a portable MRI that's been developed out of the United States. I'm not sure if we can do complex things like arteriospin labeling and so forth on that. I'm not sure the magnet's powerful enough, but you know, hopefully as advances continue, you know, we'll be able to look at that. We are getting MRIs in these people once they've stabilized and the bolt is out, so it won't be at the same time that we have the neurophysiology in place, but we have in about 80% of the people now, we are getting MRIs about day 5 to day 7 after the initial injury. So those are definitely things we're looking at. Unfortunately, our magnet at VGH has historically only been a 1.5 T, so we're, you know, it's not really powerful enough to do the more advanced types of imaging, but I believe the hospital's just bought a 3 or 7 T MR, so definitely something to look into.
Awesome. Well, I think for the sake of time, we'll have to stop there with questions. Mip, if you see anything in the chat, feel free to respond. Yeah, all right. I think I am up. All right, so I'm going to present just a small part of the research program that Mip described to you today. The title of my talk is Red Blood Cell Transfusion Reduces Transcerebral S-nitrosylethiol Release and Does Not Improve Brain Oxygenation in Humans. So as Mip alluded to, our overall research question in the ICU with these patients is how might we improve cerebral oxygen delivery to alleviate the secondary brain hypoxia that we're observing. I'll just run through this quickly because it was already described, but as we know, there's multiple factors contributing to overall cerebral oxygen delivery. And the interest for this research is looking at hemoglobin concentration with the specific purpose of determining the role of blood transfusions in potentially alleviating secondary brain hypoxia in the post-cardiac arrest hypoxic ischemic brain injury patients. Now, I want to describe a little bit of background as to why we feel this question is important. We know from several decades of work by Jonathan Stamler and many of his colleagues that there's a specific vasodilator pathway based on nitric oxide mediated vasodilator activity that's important in the setting of hypoxia. So the first point here is that this molecule that we can see on the left, this SNO, or S-nitrosahemoglobin, this is a nitric oxide molecule that's covalently bound to a cysteine residue on hemoglobin. Now, upon deoxygenation, if we move over to this hemoglobin tetramer here, nitric oxide is released in the form of an RSNO, or S-nitrosethiol for short, and that this leads to vasodilation. And through a multitude of different forms of studies, including genetic knockouts, etc., it's been demonstrated that S-nitrosahemoglobin is a principal regulator of hypoxic vasodilation. Now, for transfusion, where this becomes relevant is that if we look at the concentration of S-nitrosahemoglobin, on the y-axis here, it's plotted as the moles of S-nitrosahemoglobin per mole of hemoglobin tetramer. Following the withdrawal of blood from a blood donor, S-nitrosahemoglobin is almost completely depleted within as little as 3 hours in this study, and it remains depleted across the entire storage duration. This means that upon retransfusion of blood, there's impaired vasodilation. So looking at data from Jonathan Stamler and his group, here we have the change in tension in, I believe, aortic rings ex vivo. And with fresh blood, we see a reduction in tension of around 20% with the hypoxic stimulus. In expired blood, we see that this vasodilatory capacity is markedly reduced. And then even in day one stored blood, so what we would consider in the critical care setting, fresh and very high quality blood, there's already a reduction in vasodilator capacity. But if we take this blood and restore the levels of S-nitrosahemoglobin, this vasodilator capacity is also restored. So these three key findings lead to the hypothesis that blood transfusions may not be achieving their full potential to improve oxygen delivery to multiple organs, but in our interest, the brain, due to a potential reduction in vasodilator capacity. And so we wanted to at least start to observe and describe this in the critical care setting before we move on to potentially trying to deal with this problem. So as Mip described, we conducted this study in the patients with the invasive neuromonitoring. The data I'm going to present today is primarily focused on the partial pressure of brain tissue oxygenation. And then we have some preliminary data on individuals that have also underwent the microdialysis and cerebral blood flow catheter measurements as well. And I'll present a little bit of data on cerebral oximetry. And again, this is all sent into ICM Plus and integrated together for analysis. So looking at the results of our study, as expected, following transfusion at each time point 2, 4, 6, 12, and 24 hours, following transfusion, there's an increase in hemoglobin. So the effect that we wanted was observed, and this led to a similar increase in the arterial oxygen content. And this, of course, was expected. Looking at the neuromonitoring findings, what's the actual impact of this on brain oxygenation? First, I'm going to present the regional saturation of oxygen data from the cohort that was assessed with cerebral oximetry. And we can see that at each time point following transfusion, there's no change in the regional saturation of oxygen. Now, if we look at the patients that were assessed with invasive neuromonitoring, we again see that there's no change in the partial pressure of brain tissue oxygenation following the blood transfusion. So red blood cell transfusion does not seem to have a beneficial effect on brain oxygenation. And that seemingly is puzzling based off the fact that we know hemoglobin concentration is important in increasing cerebral oxygen delivery. but might be explainable based upon those prior results that I explained related to the vasodilator capacity of stored blood. So we wanted to try to assess this, and we did this by assessing nitric oxide species in the arterial circulation, as well as Mip described, taking our paired arterial and jugular venous bulb samples to look at changes in nitric oxide species across the brain and infer if this is indicative of potential uptake or release of important vasodilators. So here, I have our results for the arterial concentration of plasma nitrite, and we can see that, and we only have one time point here, pre to 12 to 24 hours post transfusion, there's no change in the concentration of plasma nitrite in the arterial circulation. Now, if we look at arterial plasma nitrosithiols, we can again see that there's not really any difference here as well. But if we look at the arterial to venous difference, perhaps we'll see something else brain specific. Related to the arterial to venous difference of plasma nitrite, we didn't see a difference. A positive value here would indicate uptake with the negative value indicating release, but we don't see any difference between the two time points. But when we look at the arterial to venous difference for plasma S-nitrosithiols, we see that first, prior to transfusion, there's actually a significant release of S-nitrosithiols across the cerebral circulation. So this is analyzed with a one sample t-test. And then when we look between the two time points, this difference is actually reduced, and there's no significant release anymore following transfusion. So red blood cell transfusion was associated with the reduction in the net release of S-nitrosithiols during cerebral circulatory transit. Now, as I said, in a small subset of patients, and this is in our ongoing cohort, we also collected at the same time data on cerebral blood flow, and I'll present that here. So we have 3 patients, each individual line here with the dots representing separate time points, and then I have the bars behind just for the mean of the group. And I haven't performed any statistics here because it's still very preliminary, but we can see that generally speaking, there's quite a large reduction in cerebral blood flow following the transfusion. When we look at cerebral vascular resistance, we see a large increase. And then finally, when we look at cerebral oxygen delivery, while it's a bit more variable, the overall trend is that there's definitely no improvement and potentially a reduction in cerebral oxygen delivery as well. And so these data, they suggest at this point that red blood cell transfusion reduces cerebral blood flow to an extent that might actually be reducing, oxygen delivery. In other words, the reduction in cerebral blood flow is greater than the increase in arterial oxygen content, because we do know in the normal setting of healthy individuals that increasing red blood cell concentration would reduce cerebral blood flow, or reducing red blood cell concentration would increase cerebral blood flow. There's an inverse relationship between the two that typically leads to the maintenance of a stable cerebral oxygen delivery. But in this case, it's leading to a reduction in cerebral oxygen delivery. So what I want to do is try to tie this together and explain what our operating hypothesis is as we move forward and conduct more studies. So if we look at the arterial circulation, we have our oxygen level, we have our oxygen bound to red blood cell, and then our S-nitrosate hemoglobin molecule. As our red cells move through the capillaries and oxygen is offloaded, this leads to the oxygen-dependent release of S-nitrosate hemoglobin, where S-nitrosithiols will travel to the vascular wall and elicit vasodilation, but we're picking up potentially also this increase in the venous circulation. And so in the normal setting, we have a higher venous concentration of S-nitrosithiols than in the arterial circulation. And the idea is that this is due to release from the red blood cell. But in the setting of transfused blood, the S-nitrosate hemoglobin deplete blood is likely displacing reducing the overall concentration of S-nitrosate hemoglobin that's circulating, impairing the release due to the lowered concentration, and then eliminating, as we observed, a reducing RSNO levels in the venous circulation. And this is potentially leading to a reduced vasodilator stimulus and the increase in cerebrovascular resistance that we observed. So moving forward, our hope is to better understand how transfused blood is impacting nitric oxide levels, the release of nitric oxide across the cerebral circulation, and the impact on cerebrovascular physiology with the invasive neuromonitoring. So with that, I of course have many people to acknowledge and say thank you to, and I'm happy to answer any questions. Oh, Patrice has a question. Would it be interesting to look at ATP release from red blood cells? Definitely. We don't have the capacity to do this. In the past, I've reached out to a few different labs that have published papers on this. And ultimately, the general response that I've got is that they've stopped doing it because they find it exceptionally complicated. And it's not something that they've felt super comfortable moving forward with. So while I would love to do this, I'm not exactly sure how to do this moving forward, but it would make a lot of sense physiologically to look at this because there's also that ATP release as well. All right, well, with that, we'll move on to the next talk. What I'll mention as Tony gets his slide set up here is that we're going to pivot a little bit, and the two talks are going to focus on cerebrovascular physiology during the setting of hyperthermia and hypothermia. And the reason why we wanted to do that in this seminar is, as Mip mentioned in his presentation, recently came out with the Targeted Temperature Management 2 trial this summer that therapeutic hypothermia may not hold the therapeutic promise that it was once thought. Originally, there were some studies published in around 2002 showing that cooling patients to 33 degrees was beneficial. It was subsequently demonstrated that there was no difference between cooling between 33 and 36 degrees. And then the question became, is this a beneficial effect of cooling? Or is it simply that because we're avoiding fever or hypothermia in these patients that we're seeing a beneficial effect? And so in this recent trial, their control group was just keeping patients under 37.8 degrees. And they showed that 37.8 degrees or less compared to cooling showed no therapeutic benefit. And so I thought it might be interesting for us to look at this through a physiologic lens and think about how what we know about heat stress and cold stress in the healthy human setting might be relevant to these findings and future research in that area of targeted temperature management moving forward.
Great, thanks, Ryan. I'll just give a brief introduction for Tony here. I had the privilege of working with Tony back when he did his PhD at University of British Columbia, Okanagan and Kelowna. Now Tony is an assistant professor at the University of Windsor. He's a terrific scientist and colleague, and it's always a pleasure collaborating. And probably maybe a little bit more impressive than Tony's scientific achievements, I've heard he's also an aspiring amateur ping pong player. But I think today we'll focus on his scientific work. Take it away, Tony.
Okay, thank you, Hannah. It's probably a good thing that we don't focus on my table tennis skills. So yeah, I'm excited to give this talk today, and it actually blends in nicely to Mip's keynote presentation. So I'm excited to present some of the preliminary work that hasn't been published on that just yet. So yeah, and again, I'd like to thank the organizers, Patrice and Caroline. What you guys been doing is just absolutely fantastic, especially during this pandemic, really to keep the wheels turning on cerebrovascular research. Okay, so today I'm going to present very quickly. Ryan said that I have to keep this talk within 10 minutes, so I'll try to be very quick. Two papers that are published, and then really I want to focus on the manuscript in preparation. So this is hot off the press, and it blends in nicely, like I said, to previous talks from Mip and Ryan. So starting off with the cerebral metabolism, first let's give a very brief overview of the basic control of brain blood flow. Now, you guys already know this, I'm going to be very, very quick. We know that the cerebral blood flow is impacted by neuronal activities and metabolism. Blood pressure, of course, will impact the cerebral blood flow. Sympathetic regulation, less so is known, but we know it does have an impact. And then, of course, the arterial blood gases, so carbon dioxide, increase arterial carbon dioxide tension, you'll increase the cerebral blood flow, you decrease the carbon dioxide, you decrease the cerebral blood flow, and oxygen as well. So importantly for this talk is that the hypothermia will actually impact every single one of these sort of control mechanisms for the cerebral blood flow. And specifically, I am really interested in the neuronal activity. So if you look at the cerebral metabolism, it really hasn't been known what would happen in hypothermia in vivo in humans. So we always fall back on the Arrhenius plots or the theoretical Q10 effect. Now, this is just in isolation. So this is a theoretical estimation of what would happen to the cerebral metabolic rate of oxygen on the y-axis as you change the cerebral temperature. And that's assuming a Q10 effect of 2, which is sort of your average for biological activity. But again, this is usually in isolated models and in animal models. So with this, a 2-degree increase in cerebral temperature should correspond to an increase in the cerebral metabolic rate by about 20%. But again, this is the intact model. We don't know what happens in humans, and especially because we know that in humans that become hot, you become hypocapnic. You have a respiratory alkalosis response or a heat-induced hyperventilatory response, and then a subsequent reduction in the cerebral blood flow. So you have a reduction in the convection, if you will, as Ryan is termed it, I suppose. So the question is, does that impact the cerebral metabolic rate of oxygen? Okay, so that was the main question for this study 1 here. The experimental design, we took healthy men and we warmed them up in this water-perfused suit here. Heating time is approximately 1 hour to achieve an esophageal temperature of +2 degrees above the baseline, or an absolute of 39.5 esophageal temperature. So this was Mip's handiwork here. This is me. We couldn't, based on his, I guess, best wishes, he suggested we don't put an indwelling brain probe in, but we did have jugular venous catheters as well as an arterial catheter. So alongside that, we also changed the arterial carbon dioxide tension using dynamic end tidal forcing, basically to restore the CO2 tension back to normal thermic values, which is around 40 millimeters of mercury, to really control for the effects of the hypocapnia and therefore the cerebral blood flow. So like I said, we had arterial measures and jugular venous measures, and then we also measured the cerebral blood flow. So you combine that together where you take the difference of the arterial and jugular venous oxygen concentrations times that by the cerebral blood flow, and you can then calculate the cerebral metabolic rate of oxygen. So this is just a basic Fick principle. Okay, so first starting off with the cerebral blood flow, what we see is, as we'd expect, when we keep people up to about 2 degrees Celsius above their normal thermic baseline, we get about a 20% reduction in the cerebral blood flow. And there's nothing new about this. This has been known for several years. Now, when we clamp the CO2 back to the normal thermic values, then we essentially restore the cerebral blood flow back up to the normal thermic values, at least on average. And again, this is nothing new. We know that in heat stress, at least passive heat stress, the reduction in the cerebral blood flow is primarily attributable to the heat-induced hyperventilatory response. So now what about the cerebral metabolic rate of oxygen? So just to orient you to this graph on the left, we have the percent change in the cerebral metabolic rate of oxygen. On the top here, I have the arterial carbon dioxide tension, and the bottom, we have the change in core temperature. So what we find compared to 0 as we move through, as we increase our core temperature, our cerebral metabolic rate of oxygen, in fact, increases and completely in line with the theoretical Q10 effect, so the Arrhenius plot. Now, this is despite the fact that you actually have that reduction in cerebral blood flow. So as if you can recall from MIP's presentation, his wonderful presentation, you have an increase in the diffusion, so an increase in the extraction of oxygen despite the fact that the blood flow is decreasing. Now, what about when we change or we restore the arterial carbon dioxide tension? So if you look at the top there, you see that at 2 degrees Celsius, our PaCO2 is about 27.4. So that's about 10 millimeters of mercury below our normothermic values. Well, when we restore it back to normothermic values, but maintain heat stress, we really see no change in the cerebral metabolic rate of oxygen. Granted, it becomes a little bit more variable, but essentially no change. And of course, that suggests that the cerebral metabolic rate of oxygen is increased by about 10% with each degree Celsius increase in core temperature, confirming that Q10 of 2, and irrespective of the respiratory alkalosis or the subsequent reduction in the cerebral blood flow. Okay, so there's one other thing we did measure in this study, and that was the oxidation and inflammation coming from the brain. And one thing that stood out, at least to me from that, was you actually had where there's a trend for an increase in interleukin 6 from the brain. So the next question was, well, is this level of heating, is it problematic for the cerebral parenchyma, and does it open the blood-brain barrier? Because we know that interleukin 6 is a potent modulator, that cytokine is a potent modulator for cerebral inflammation and blood-brain barrier opening. So that was the next study we did, exact same experimental design with MIP's handiwork here. The only difference is we had no CO2 clamp. Now, I'm just going to briefly go over this because I stole and adapted this slide from Ryan, and MIP has already gone over it. But we essentially used the cross-brain kinetics to determine or to measure markers for parenchymal damage. So we have neuron-specific annulase, which is the marker for neuronal cell body injury, and then tau and neurofilament light chain for markers of axonal injury. And we also measured the S100 beta, which is a relatively validated marker for blood-brain barrier permeability. Essentially, it's a big protein that is primarily released from the astrocytes. And in an attacked blood-brain barrier, it doesn't get into the circulation. If that blood-brain barrier opens up, then it allows the large protein S100 beta to cross the blood-brain barrier and get into the circulation. Okay, so I know I'm moving quickly through this, but that is okay. If you have any questions, again, you can just fire it in the chat after and we can talk offline. So I just want to quickly go over some of the results here, which were actually quite, I guess, surprising in the fact that despite the fact we had the increase in IL-6, there was no noticeable change in S100 beta. So I'm just presenting the cross-brain kinetics where the negative denotes a cerebral release and then a positive would denote a cerebral uptake. So S100 beta, there's no indication for acute blood-brain barrier opening, at least, again, I should reemphasize, this is healthy men at about 39.5 degrees Celsius. We can't say this would be the same thing for a pathology, but at least in health, the blood-brain barrier seems to be intact. Now for neuron-specific annulase, again, no significant findings. There's no change in the cerebral release. And likewise for axonal injury, the neurofilament light chain didn't change, as well as the tau protein. So really nothing to take home from that. Well, I guess one of the take homes is that this level of heating is, at least from these data, it's safe for the brain parenchyma and for the blood-brain barrier. So the title of the paper that was, I should acknowledge my master student, Brooke Shepley, she wrote it up. The negligible influence of +2 degrees Celsius increase in core temperature on the blood-brain barrier permeability and cerebral parenchymal integrity. Now, one thing that I really want to emphasize here is that this was the hypocapnic or poikilocapnic, I should say, heat stress. In other words, we allowed people to hyperventilate as they normally would in heat stress. So that raises the question, is the hypocapnia in heat stress protective? Right? We didn't see any noticeable changes in blood-brain barrier permeability or our markers for neuronal damage or axonal damage. And so to really glean this, I'm going to look at it through the release of microvesicles. And typically microvesicles in so endothelial-derived microvesicles in high concentrations are associated with a pro-inflammatory phenotype. So my question was, does restoring the PaCO2 to normal thermic values in heat stress impact the presence of these circulating endothelial-derived microvesicles? Okay, so the next series of slides I'm going to show, this is, like I said, hot off the press. I made these figures up last night, so just bear with me here. And I apologize I didn't put the individual values. I do show individual values for exchange. So first, if we look at the CD62E, which is Eselectin, right? So MIP had a great present. I didn't see those data before, and I was quite excited about that. With the venous activators, so CD62E is considered your activated phenotype of endothelial microvesicles. And then we also looked at the CD31 positive, CD42B negative microvesicle, which is considered an apoptotic phenotype. So when we look at it just coming from the jugular venous circulation, there's really no discernible change when we move from baseline to heat stress, and then the same level of heat stress with the inhalation of carbon dioxide to restore the arterial carbon dioxide tension back to normal thermic values. Now, notice I have a really high y-axis here. The scale of the y-axis is quite high, and that's because we need to compare it to the arterial side. So when we look at the arterial side, we see a very marked noticeable increase in the both activated microvesicles as well as the apoptotic phenotype, especially the apoptotic phenotype. Now, I must admit, I've been sitting on these data for a while, just because when I first saw this, I thought, wow, there has to be a mistake here. There's about a 400-fold increase in the arterial apoptotic microvesicle release when we restore the carbon dioxide to our normal thermic values. So I went back to the flow cytometer tech. I went back to all the samples, and there's nothing can be, everything was run in the same batch, in fact. So there's no, well, at least that I can see, methodological error in these data. And when you look at the exchange kinetics, because you have such a high arterial uptake or release of these microvesicles, you in turn get a cerebral uptake. And so the positive is denoted by an uptake, of course. And you get an uptake of both the CD62E, which is Eselectin, or activated microvesicle, as well as the apoptotic phenotype, or especially the apoptotic phenotype, I should say. So just in summary, I guess, restoring the partial pressure of carbon dioxide to normal thermic values, it caused that robust increase, and then cerebral storage. So a few considerations here, this notable increase following the CO2 restoration, it may signify a pro-inflammatory response that, of course, is likely to exacerbate heat-related illness, because we know that in heat stroke, it's first, of course, the cytotoxic effects of heat, but it's secondary, or the major pathophysiology is secondary to the pro-inflammatory response of your body to the cytotoxic effects of the heat. So really, our microvesicles driving this, or in part helping drive this. Now, of course, MIP would know much better here, but I just wanted to put this out for him. Clinically, in heat stress patients, so regardless of etiology, maybe even sepsis, might they fare better under moderate hypocapnia, or at least make sure that they are not hypercapnic? And it could be a lesson learned. So I'm just going to kind of leave it here. It could be a lesson learned from, in fact, our heat acclimation response, our normal heat acclimation response. And this always kind of stumped me, because we always consider the hyperventilation and heat stress as a maladaptive response. But when you heat acclimate someone, despite the changes in the thermoregulatory improvements, blood volume improvements, the orthostatic status improvements, they don't change, or you don't change your hyperventilatory response. So that is conserved in heat acclimation. So might it in fact be protective? Is there some evolutionary protection, and could it be explained by the microvesical release? So nice and quick. I hope I kept it under 10 minutes. That's it. And I'm kind of leaving you there with those preliminary data. Again, very hot off the press. And I'm happy to take any questions now or, of course, after this presentation.
Hannah, could I ask a question?
Yeah,
Craig.
absolutely. Go ahead, please, Craig.
Thank you, Tony. That was wonderful. We sure enjoyed that. Any thoughts on just giving a CO2 load to normothermic individuals and looking at this EMV and to see whether there's any effects of the CO2 independent of the heat?
Yes, I do have those data, Craig, and I tried to get them out for today. But I must admit, I was a little crunched for time. But I'll definitely keep you posted on that. Yeah, I don't think you see the same thing as you do in the heat. So I think there's something happening with the heat that's kind of priming the release of these endothelial-derived microvesicles.
Great, thanks, Tony. Might have time for one or two more questions if anyone has something in the chat. Perfect. So Patrice, thanks for an interesting talk. Anthony, would you expect to see similar results in young women or older men/women, or in patients with increased core temperatures, such as sepsis patients, for example, for your studies 1 and 2?
Yeah, so that's a big caveat, of course, of these studies. They were limited to men only and healthy men. So in terms of the sex differences, to be honest with you, I can't foresee that there would be any difference, at least in healthy females. But of course, in pathology, then, yeah, we know in TBI, of course, now whether there's a manifestation of the disease or not, but any mild hypothermia is quite a problematic and ominous sign. So yeah, it's something that I don't know if we can get at ethically in a more clinical population. Aging would be interesting to look at for sure. So yeah, we have to hold these data with the caveat that it's in healthy men for sure.
That's great. And thanks, Tony. A question from Kate here. Do you think the pro-inflammatory markers could be related to the stress of restoring CO2 in an already stressed state rather than an effect of cerebral blood flow?
Yeah, I don't think it has to do with the cerebral blood flow. I think it's directly related to the carbon dioxide, because the major increase, remember, in these circulating microvesicles was in the arterial side. So yeah, I think that there are some dish studies suggesting that if you increase the CO2, you can release these microvesicles. But again, dish studies, you put anything on the dish and it basically increases the microvesicles. So they're hard to, it's hard to interpret that model, at least in the intact human. But yeah, I certainly think it's not related to the blood flow regulation per se. It's more related to the changes in pH. Or direct impact of CO2. Yeah.
And if I may, just a related comment. So your comment here about whether this hypocapnia could be adaptive response or a beneficial response to heat stress. It could also be related in different modalities of heat stress, like sauna or hot water immersion. I think, and maybe Travis will talk about this next, seeing actually a blunted cerebrovascular CO2 reactivity response could also contribute to this protective effect that you're actually seeing reduced sensitivity to CO2, so less marked changes or reductions in CBF.
Mm-hmm. Yeah, certainly. Just with the, I mean, this is very a hypothesis sort of driven presentation here, and I thought this would be a good group to send it out to. But when you do look at heat acclimation, or at least when you do look at the hyperventilatory response in heat stress, and Craig's done some fantastic work to here, it doesn't seem to impact the orthostatic tolerance. So when you restore CO2, it has no impact there. So the orthostatic tolerance is mainly related to central blood volume. And it only really becomes problematic if it becomes too severe and you start to get into the hypocapnic tetany, but that's very, very rare. And with heat acclimation, it's conserved. You still have this hyperventilatory response. So yeah, and so that's kind of, as soon as I start to put those two together, I'm like, you know, let's go back to these data. And Okay, maybe there is something going on here. Maybe there is a reason why we conserve that hyperventilatory response in heat apnea as well.
Great. And maybe one last question here from Phil. I wonder if there's any relationships between changes in AvCO2 or pH and microvesical release?
Yeah. Well, from the brain, is he talking about?
Yeah. Any changes in the AvCO2 pH gradient, I suppose.
Right. Well, you have to remember on the venous side, we didn't really get that increase in microvesical release.
This is all arterial driven.
Yeah. Yeah. So why the brain doesn't seem to be releasing it and just uptaking it. But importantly, this is uptake of Eselectin, remember. So yeah, so and just seeing Mips talk after, thinking, wow, this could be actually a sort of a pathway for the pathophysiology of heat stress, heat illness.
And then maybe, maybe to look at the severity of the alkalosis within individuals, and if that relates to within individual microvascular.
Oh, sure. Yeah. Oh, good point. Yeah. I haven't done that yet. Yeah. Again, these were last night.
Because maybe that could give an indication. Maybe the participants that have the largest hyperventilatory response, for example, are seeing a larger increase in microvascular release.
Yes, good point. Yeah, I definitely have. There's still a lot I need to do with these data. So
I'd
be happy to have you along and help.
Yeah. Cool. Awesome. I think that's good for questions. Maybe we'll move on to Travis's talk. Thanks again, Tony.
Right. Hi, everyone. Just while Travis is setting up his slides, I'm just going to introduce him. I can confirm virtual jet lag is a real thing. It's 5:30 in New Zealand, and we've been up since 3:30. So it's bear with us. So it's my pleasure to introduce the newly conferred Dr. Travis Gibbons. He completed his master's at the University of Waterloo under Prof. Richard Hewson, but he spent the last 4 years working towards his PhD in New Zealand with supervisors Jim Cotter, Luke Wilson, Phil Ainsley, and myself. And he's just completed what all the examiners said was an impeccable and exceptional thesis in which he examined cerebrovascular responses to extreme environments. I can confirm all of his experiments were extremely demanding to perform, but also to participate in. So Travis is going to present some of his data to us now on the cold brain from his PhD. And also, Travis is the winner of the prestigious Journal of Physiology's Next Top Model competition. And if you haven't seen his author bio photo for his 2020 and 2021 publications, please go and search them right now. You're in for a treat. Thanks, Travis.
Thank you for the introduction. Can you hear me, Kate?
Yeah, good to go.
Cool. So the talk today is looking at cerebral blood flow responses to the cold in the healthy conscious human. Why study the cold? Cold is common in real world environments, whether you're in a Dunedin flat or up here in the mountains. It's common in therapies, for better or worse. And as Mip was alluding to a little bit, it's quite common in critical care, at least for the last little while. And it offers unique neuroprotective potential, some of which we probably haven't harnessed to its best ability as of yet. And we can have some interesting anecdotal evidence from these recoveries from people who have been immersed in water for up to 66 minutes. And this is where the comments come from, like nobody's dead until they're warm and dead, because of these people who are seemingly brought back from the dead if they were cold. Why is the cold brain relevant? We rely almost exclusively on behavioral thermoregulation to protect ourselves from the cold. So as all of you know, when you start to feel cold or you sense a cold threat, what you do is you just get out of that environment. You don't really rely on your physiological responses. And in order to interpret or perceive a cold threat, you need to have a functioning brain. And for that, it's important to understand the regulatory responses of the cold brain. And not only that, understanding how the brain responds to the cold will allow us some further mechanistic insight into how we can potentially harness it to provide cross tolerance to other stressors like hypoxia, which is used in clinical care. How do you cool the human brain? Because the brain has so much flow, it's impossible to really isolate the brain and cool it specifically, so you have to cool the whole body. You can do internal cooling methods, such as bolus injections of ice saline or ingesting slushy, cold slushy. But what we use in the lab and what's commonly used in clinical practice is surface cooling. And the key here is you often need high thermal conductance medium in order to really suck the heat out of people. You can have people standing in cold air for hours and not budge their core temperature. So if you want to cool someone quick, you really need a medium with high thermal conductance. This is steel here in the cold tub. It's a very elaborate study we did in Phil Ainsley's lab. Something you should also know is that there's huge variability in how people cool. I'm not sure if this is the same in critical care, but if we cool a healthy conscious human, there's basically a wide spectrum of phenotypes. And I kind of characterize them as a metabolic compensator, someone who shivers aggressively and really protects their core temperature. A tolerator, I don't even think that's a word, but a tolerator would be someone who just allows themselves to get cold and they don't really fight it. And then an insulator is someone who uses their insulation to prevent cooling. So this is just two examples. The timeline's about the same. This is about 30 minutes in here. So this subject here cools by close to 2 degrees in 30 minutes. And this isn't about 8-degree water. When you pull them out of the water, they just try to heat up sometimes they get a 3-6-fold increase in metabolic rate just from shivering thermogenesis. So it's almost impossible to keep these people or this phenotype cold once you get them out of a water bath. Whereas subject B here would be more of a tolerator. It's kind of like cooling a piece of meat. Slowly but surely you cool them down, pull them out of the water here, and they don't really warm up. So a very minimal shivering response. Subjects like this are easy to collect data on because they're not shivering and they're not heating up very quickly. Subjects like this are shivering vigorously. It's almost like a state of exercise. So something to consider when you're attempting some of this research. And then all these dips are swallows. So this is esophageal temperature readings here. And then this would be an example of an insulator. So just comparing these two subjects, this is about 3 to 4-fold slower rate of cooling, despite the exact same water temperature, and not even a very different anthropometry, probably 23, 25 body mass index. So you can't even really perceive a difference in insulation, but there is definitely an effect on the rate of cooling. So what happens when we get cold in relation to the cerebral vasculature? Well, the first thing that happens is you have a robust hyperventilatory response. This is an increase in breathing frequency and tidal volume. That drives CO2 down. And as Tony was talking about, this plays a pivotal role in decreasing cerebral blood flow. So this is hyperventilation, hypocapnia, and hypoperfusion are the characteristics of the cold shock response. However, this hypoperfusion is quite dependent on a lot of factors related to how we enter the water. The rate at which we enter, whether we're passively entered into the water or whether it's an active immersion and we have to swim or maintain our head above water. We can actually restore this back to baseline relatively quickly if we are active. And one of the reasons why I find cold stress so fascinating, especially in relation to the brain, is that beyond this 3 minutes or so of cold exposure, in the healthy human, we know almost nothing. No one has really studied deep body cooling and how it affects cerebral blood flow, despite it being used in clinical practice for like 2 decades. So this is some of our recent research. I haven't shown anyone this yet, so what a treat for you guys today. Here is baseline, -1 core temperature, -2 core temperature. So achieving graded cooling is a bit of a challenge. We're still trying to work out the methods ourselves. But as Tony was talking about too, with the Q10 effect, if you heat up a tissue, you increase its metabolic rate, and vice versa with cooling. If you cool a tissue, you decrease its metabolic rate. But because we have such a strong thermogenic response to the cold, we actually increase our metabolic rate. So we can see a significant increase in VCO2 with graded cooling, and this is dose dependent. Very different from exercise. If you look at this as mild exercise, which it kind of is in a way, a lot of muscular contraction, there's no change in heart rate. And oftentimes we see decreases in heart rate with mild cooling. Especially with what we do to the skin. If we manipulate skin temperature with a cold core, we can get really low heart rates. So we have subjects here down in the high 20s sometimes. And blood pressure increases, largely driven by increases in systolic blood pressure. Blood pressure is tough to measure when you have a cold shivering subject. So this data is either from automated Tango cuff. Obviously, you can't use a Finipress when you have no blood flow going to your hands, or intra-arterial lines. So you'd think this increase in blood flow or blood pressure, there's also about a 20% increase in cardiac output here, would favor an increase in cerebral blood flow. But what I'll show you next will give you a better explanation of the cerebral blood flow response. So what we find quite interesting with cooling is that there's this robust hyperventilatory response. And you might expect this because we have an increase in VCO2, so this is metabolically necessary. However, it is disproportionate. So this is the second study we've done where we've seen this cold-induced hyperventilation. And similar to what Tony was talking about with the heat, this response is largely what appears to be driving decreases in cerebral blood flow. And we see the same thing in heat, where we see quite a lot of differences in individual variability in their hyperventilatory responses to the cold and the heat. But this magnitude of hypocapnia that ensues tends to drive the decreases in cerebral blood flow. So this is a second study where we did this. This is 14 subjects, healthy males and females. We did this previously in Canada with Phil's group, and we saw a more pronounced hyperventilation, more pronounced hypocapnia, and a much greater decrease in cerebral blood flow. So this here is about 15% decrease in cerebral blood flow. When I did this previously, we saw about 20 to 30% decrease, and it appears to be driven largely by the hypocapnia. But when we think about cerebral oxygen delivery and relating this to its potential neuroprotective benefit, what we also see with cooling is this graded hemoconcentration. And by increasing arterial oxygen content, you will protect yourself from this hypoperfusion. And that's exactly what we saw in this second study. Cerebral oxygen delivery was maintained despite cerebral hypoperfusion by this mild hemoconcentration response. There's no change in cerebral oxygen delivery. And that's in contrast to when we did this a couple of years ago in Phil's lab. We saw about a 10 to 20% decrease in cerebral oxygen delivery when we cooled people at sea level and at high altitude. And that was just driven by a greater decrease in cerebral blood flow. And then something else that's kind of interesting and relates to Ryan's stuff as well is that what we did in Phil's lab over in Canada is that when we cooled people down, this was quite a rough study. for the subjects. Some of them are online here, so thank you for participating in that. We brought people down 1 degree core temperature, and then we made them acutely hypoxic. So this is a percent change from normothermic normoxia. So coming down here, we're at about a 30% decrease in cerebral blood flow. And then when we made them hypoxic, bringing their SATs down to 82%, cerebral blood flow returned basically back to baseline. In the hypoxic cerebral blood flow response was about 7 or 8-fold greater than it was when we were normothermic. So you're much more sensitive to a hypoxic stimulus when you're cold relative to when you're normothermic. So in summary, when we think about deep body cooling and its relationship with the brain, cooling causes hyperventilation. The hypocapnia that ensues decreases cerebral blood flow. Hemoconcentration protects cerebral oxygen delivery in the face of this decrease in cerebral blood flow. And then with concurrent hypoxia, cerebral vascular reactivity to this hypoxia further protects cerebral oxygen delivery. That's it for me. Just like to acknowledge my supervisors, Jim Cotter here, Kate Thomas, Phil Ainsley, and Luke Wilson. All of them at one stage were participants in these kind of torture studies. And also these two guys here who helped me a lot in New Zealand and from afar, Gary Mack and Jerry Dempsey. That's it. Thank you.
Thanks, Travis. Any questions there in the chat from anyone in the audience?
Can I just ask a question? It's taking me too long to type.
Yeah, go.
Do you think that the increase in reactivity with the cold stress, the hypoxic reactivity, does that seem to be related to other factors like changes in mean arterial pressure? Or do you think that it's like a change in the actual like vascular sensitivity?
Yeah, that's a good question. I think part of it will be related to the fact that you're in this hypertensive state, and potentially if you do have a reduction in conductance or a vasodilation due to hypoxia, you've just got a higher driving pressure pushing it in. Yeah, we could easily look at that data. We have it, but I do think it is probably related to the pressure in some way.
Cool, thanks.
There's a question here for you, Travis. Does weight or the amount of adipose tissue affect the findings on your first couple of slides?
Adipose tissue definitely does. The thing that I find interesting is that sometimes the difference in adipose tissue, especially subcutaneous fat, is almost imperceptible. You can tell there's an insulator by someone who just doesn't cool down, but you don't actually, it's not always extremely apparent, if you know what I mean.
Cool. Anyone else got some comments out there?
Like Mips.
Can I just ask a quick question? I'm sorry, much like Brian, I'm slow on the typing. Really cool stuff. Thank you very, very much. My question kind of relates more to sort of how we can be better as clinicians. What is it about, clearly hypothermia is protective for neural tissue, but why are we not seeing some of the perceived physiological benefits translate into improved clinical outcomes? Like what are we doing wrong in our trial designs and so forth? And then my second question, just a slight follow-up, is how do I also apply to become a model for JPhys?
I guess I'll start with number 2. You just got to take a lot of time with the photo shoot before you submit that author photo. But for your first question, I really don't know how you should approach it in the clinical setting. I know that there are some theories about if you have a cold water drowning, what's protecting the brain, and this is one of, I think, Mike Tipton and Goldman's theories, is that you aspirate the water and you can cool your carotid artery blood temperature by about 8 degrees in 2 minutes. So you're rapidly cooling the brain while your heart is still pumping, and you probably haven't had the huge hypoxic insult on the brain by the time the brain is cold. So I think it could be a timing thing. There's a lot of different factors that come into play, as you well know, with timing and depth and rewarming processes that are quite complicated. And as you and Ryan showed with your kind of different cohorts of hypoxic brain injury, understanding the nuances of each patient is probably quite important in how you try to approach therapeutic hypothermia. And I think it's an area that's ripe for further research, just understanding the mechanisms before jumping in and just trying it almost.
Here's a question here from Tony Trav. With the cooling in clinic, are you just delaying the outcome of the injury without necessarily fixing the underlying pathology? I guess maybe that's directed at MIP potentially.
Maybe MIP can give a go at that. He knows it way better.
Yeah, I'm not sure. I think, Travis, you hit on something really astute that kind of resonated with me in terms of why we're not seeing the improved clinical outcomes with respect to hypothermia in clinical trials. I think the probably a big factor in that is from the timing of injury to the institution of hypothermia. Like, we've had numbers of cases, innumerable cases of patients who have arrested from hypothermia for 3, 4, 5 hours in some cases, or 3 hours being the most, but with successful outcomes. Whereas what we do I think how we use hypothermia clinically otherwise is the patient who is normothermic, who arrests from, let's say, a myocardial infarction, they're normothermic, and then our median time to getting these people down to hypothermia is like 6 to 8 hours. Pragmatically, it just takes that long to institute hypothermia, and so I think the question is very different. Is hypothermia neuroprotective? My personal opinion is yeah, it probably is. But pragmatically, how we use it in the clinical setting is that, like, that 6 to 8 hour delay in taking a normothermic person and putting them into hypothermia. How we use it in clinically, it doesn't seem to work. So, I don't think, unfortunately, these trials are good. Like TTM2 was a very good trial, but I think it's led to throwing the baby out with the bathwater. It's relatively unfortunate because I do think there are important lessons in there.
That's interesting, Rip. I think it just adds on to what Travis was saying with the, I guess, surviving. I guess if you survive drowning, it's usually with rapid cooling, especially young kids, because they can cool so fast. So yeah, I suppose the timing that you get the patient to be cooled is really important.
And some of this came out like with Stephen Bernard's initial studies out of Australia in the 1990s, and Austrian study, the Haka trial. They were smaller, and there was other confounding there with respect to blinding and all that. But the hypothermia groups did better. But they acted upon cooling the patient quite rapidly, and so whether or not the flip side of the TTM2 trial, very large study, 1,700 patients. It's a very pragmatic study, very generalizable. In not all places it's near to impossible to get people cold very quickly post arrest for a number of factors, but if there was another way that would not expose the body to the systemic effects of hypothermia, but selectively the brain, then I think it might be might be more reasonable. I also think in the injury setting, we tend to view all patients the same. The pathophysiology is the same, the phenotypes are the same, whereas I'm not sure that's the case. And so the other question is there a select group of people that we should be doing this in? And then secondarily, how do we identify them?
Yeah. Sorry for my ignorance, but what about sort of prophylactic cooling in let's say, heart transplant or something? I know that's quite commonly done, where you're actually cooling before you provoke the injury.
Yeah. So like for the actual transplanted heart?
Right. Well,
Yeah.
not for the heart itself, but for the patient.
Oh, for the patient, so there is a practice of deep circulatory arrest in select cases, particularly for aortic arch surgery,
Right.
where the both carotids and the entire cerebral circulation is going to be cut off transiently for half an hour, at most maybe an hour, where patients are at times taken down to 18 degrees with cardiopulmonary bypass. So that is certainly, and I do think that bringing temperature down upon return of spontaneous circulation or reperfusion, whether it's unclamping the carotids or return to spontaneous circulation, is an entirely different physiology and pathophysiology than return of like reperfusion and then 8 hours later getting somebody to hypothermia. Like that's, I think they're fundamentally different. And regrettably, I think a lot of the clinical world would take the lessons of TTM2 and say, well, hypothermia doesn't work for anything, and it's not neuroprotective. The scientists don't know what they're talking about, which I think is fundamentally not true.
Yeah. Interesting.
I think the plan was to open up questions to any of the speakers at this point. So if there was anyone else out there with questions for any of the talks that have gone before, feel free to jump in now.
I have a question, if I may. So my question was mainly related to one of the upsetting results on NIRS. I was hoping that it could be more specific. What was the limitation? It was simply the technique that was missing, where the results were not the capacity to match the results with the partial oxygen pressure. So if you can please expand a bit that part of your study. Thank you.
Of course, yeah. We plotted bilateral cerebral saturation or near-infrared spectroscopy, the regional saturation of oxygen against ppTO2, against mean arterial pressure, against CPP. Failed to find a relationship. So question is does NIRS reflect cerebral hemodynamics in ischemia reperfusion setting, like hypoxic ischemic brain injury? I'm not sure that's the case. The reason I say that is, some of the diffusion limitation literature I showed, where there appears to be some abnormalities in the exchange of oxygen between the vasculature and the parenchyma, because NIRS is taking the integration of hemoglobin saturation in the arterial tree or the arterioles and the venules. Well, the only way then it should be an accurate reflection of cerebral blood flow would be if that exchange of oxygen was normal in the setting of hypoxic ischemic brain injury, and we now are increasingly knowing that it's not. So if the exchange of oxygen is not abnormal, well then, and the coupling between convective oxygen delivery and brain oxygenation is not normal, then I think it fundamentally means that the technique in and of itself does not accurately, in a disease setting, reflect cerebral blood flow. What's very interesting about NIRS is repeatedly authors have shown that it in the setting of post-cardiac arrest, there's an abrupt increase in the RSO2 value, whatever that means, upon return of spontaneous circulation, so upon the heart restarting. But in the post-resuscitation setting, no matter which way you look at it, doesn't that the values observationally don't have any sort of links to really clinical outcome, favorable neurological outcome, and so forth. And so I think that probably that longitudinal pathophysiology, like diffusion limitation of oxygen delivery, probably starts to take place hours and days after return to spontaneous circulation, typically when people have used NIRS. And so if there's not, if the exchange of oxygen is not accurate, I don't think the NIRS value probably represents much in a disease setting. Ryan's also published some data as well, even in healthies, that the NIRS value doesn't appear to have a really robust relationship to overall cerebral blood flow even. So not to say, and finally, I think there are limitations in the technique in and of itself. There's, widely reported literature, NIRS picking up, and we have literature as well, NIRS picking up a number, an RSO2 in neurologically brain dead patients as well, who have absent cerebral blood flow. So in the clinical setting, I'm not really sure how to use it. We also plotted it against PbTO2, no relationship. So we had high hopes, but sometimes a negative finding or a series of negative findings are very important.
Thank you. Thank you.
Well, if there is no other question, Caroline and I would like to thank all the speakers for this fantastic seminar. It's definitely because of people like you, dedicated people like you, that this seminar is successful. So again, thank you very much, and see you all in November. Those next speakers will have a lot of pressure on their shoulders. Thank you very much.